grow just our target organisms, or we at least get that target group smaller. So to do this, we need a couple of different types of media. So when we work in the lab, when we get like a urine sample or a throat swab, we're going to set up some different types of plates. We're going to set up some that are called selective media and some that are called differential media. Sometimes the same type of media will work together. So sometimes these are combined. And what I mean by that is that sometimes we're going to have media that is selective and differential, which is really nice because then they go hand in hand. So selective media means that it's going to have ingredients that only grow target organisms. So target organisms could be pathogens, but they could also be maybe a particular organism we're looking for in the environment. If we're looking for organisms that feed off of petroleum, we could design media that has petroleum elements inside of it. So usually these are pathogens when we're talking about clinical use, but they don't necessarily have to be, all right? So usually they're pathogens for our use. So with the way that we grow these target organisms, it's because they're going to have selected food types in them. So they're going to have selected foods. So we talked about before with nutrient agar how it had carbs and lipids and nucleic acids and proteins. Add a little bit of something for everybody. I mentioned it was kind of like, a salad bar or something. These don't have that. They only have selected food. So this could be like certain types of sugars. And if my organism can't eat that sugar, then what's going to happen to them?
It'll die.
They're going to die. Yeah, so I want to get rid of them. So this is a way I can get rid of background noise. All right, so I'm selecting out the organisms. I can do certain food. I can do maybe selective ingredients, selective agents is what we call them. These could be things like certain chemicals or antibiotics, which will kill off their background bacteria. Another one that we can do is dyes. It turns out that there are certain dyes that will kill gram-positives, sorry. The reason for that is because gram-positives have to build those thick peptidoglycan layers. When we have dye in a lot of different types of media, the dye gets in between the peptidoglycan layers and makes them kind of slippery and they fall apart. So it makes it a little bit harder to grow them. So that would be an example of a selective media. It has something in it that makes it select certain organisms. Now we also can have differential media. Differential media is when we see a visual difference between similar organisms. Now, these similar organisms could have the similarity of being like all gram-negatives or all gram-positives or even the same genus. So for example, we're going to work with some media today that is going to separate out Staphylococcus aureus from Staphylococcus epidermidis. And we're going to see a visual difference between the two. Now, a lot of times these, again, kind of similarly to our selective media, can rely on metabolism differences. So we're going to learn that we can adjust their food, just like kind of what we were doing up here, but even among Staphylococcus, Staphylococcus aureus will eat certain things that Staphylococcus epidermidis won't. So they kind of go hand in hand. So the beauty of this is when I combine these, if I have a media that's selective for, say, only Staphylococcus, and then it even goes a step further, it's differential between aureus and epidermidis. That is great, because that's taken a lot of effort off of me. All I'm having to do is put a sample on a plate, and then I come back and read my results tomorrow. And after 24 hours, I can tell what I have. Now, the beauty of this also is if I do a good job, I may even be able to not have to do a gram stain right away. Because, for example, if I'm growing something that can only be Staphylococcus, what reaction does Staphylococcus have on the gram stain?
Gram positive.
Gram positive. So this will even help me make sure that I have the right gram stain results. So I can even wait a day to get these growing, and then I can do the gram stain from these plates that I'm setting up when I've isolated out a lot of the background noise. See the beauty of that? Kind of goes hand in hand, right? Okay, now we also have another type of media. I'll put it up here just to have it in our bag of knowledge. Oh, like, where is that? I can feel my pen, it's hiding in my dress armor. Okay, another type of media is something called enriched media. We don't have any of that today, but I will put it up here because whenever I talk about those two, I like to talk about enriched media as well. Enriched media is a media that will encourage, and that's the key word, encourage growth of a target organism but not inhibit. And that's a key there, but not inhibit. Background. Flora. So the key for this guy, let's say that I think, where we use this one a lot is with things like salmonella. Let's say I think a patient who's having stomach pains and upset stomach and diarrhea has a salmonella infection. So I will put their fecal sample in an enriched media. Now this enriched media has the exact favorite food of salmonella. So this would be like me getting a broth full of, I mentioned before, like Cheetos and Starbucks, all right? That's like my dream right there. So this is like giving them exactly what they want to eat. That being said, there's no antibiotics, there's nothing in there that will kill other background. And that's important because what if we're wrong and the patient doesn't have salmonella? So we're encouraging if there is salmonella present, it will grow faster. But if it's not present, we'll still grow. Maybe they have O157, maybe they have Shigella, maybe they have something else. So we'll still be able to grow that organism, we're not inhibiting it. It just won't grow as fast as our salmonella target would. Does that make sense? So we're like, we have an idea, we're using this media because we have this idea, but if we're wrong, no harm, no foul. We can still find the pathogen that's hiding in there. So that's the beauty of the enriched media. Now, today we're going to be setting up mannitol salt agar, EMB, McConkey, and nutrient agar. Now, we've worked with nutrient agar before in Lab 8. Was nutrient agar selective or differential in any way, or was it just a general growth media?
General.
Good. So this is our general growth media. Why do you think I want to set up a general growth media when I'm setting up these other ones? What would be the purpose? You got it. It's going to serve as my control. Again, in a laboratory setting, we would always set up some sort of control, a general growth. Usually it's not nutrient agar, usually it's something like blood agar, that we can pick pathogens off pretty fast. Blood agar, we'll talk about that a little later, we'll actually have a lab that uses it. Blood agar is usually from sheep's blood, okay? And I'll give you this little heads up. When we work with blood agar, it is not from slaughtered sheep. It is actually from sheep that are like so pampered you wouldn't believe it. They're raised in Montana. They have a ranch and they circulate the sheep. They have different colored collars and they come in once a month to get a blood donation, kind of like how humans give a blood donation, and then they're re-released back out into the fields. The reason that's important, early microbiologists like Koch and Pasteur, they tried to use slaughtered blood, and when an organism is getting slaughtered, it's high stress, and the bacteria, there's enzymes in that high stress blood, it would make the bacteria not grow. They would pick up on the stress of the animal and they wouldn't grow. Isn't that crazy? So there was some sort of cellular communication. They were kind of like, the endospores were like, no, something's bad going on. And same with bacteria. So we have these pampered sheep, they have pampered horses. They're treated really well. They, like I said, they alternate. It's like crazy. I got to visit the ranch and it was really fun. It was really neat to see. It's like, yeah, it's like something out of a magazine. You're like, are you kidding? Like, can I do this? Can I just rotate? Okay, so nutrient agar is going to be our control today, but we are going to set up these three types of media, and these media are going to be selective and differential. So we're going to see some pretty cool results. next time. I'm just gonna put S and D, so I'm writing that. Now, the mannitol salt auger is going to be, do I want to go into this now? Maybe I'll wait. I think I'll wait and go into depth on these next time when we have our results in front of us, and we'll start talking about the different ingredients. That way you guys aren't having to listen to me lecture too long. I always hate to lecture too long in lab. I feel like that defeats the purpose. But I will say this. The media that you're using today, you want to be very careful with. And what I mean by that is your bacteria are tattletales. If you set up the wrong microbe, they're going to rat you out because they're going to have a different reaction on the media than we expect to see. And you can tell me, nope, I put E. coli on there, and I look at your plate and it's all, I'm staph, and I can tell it's staph because of the fact of its reaction. They will tattletale on you. They'll be like, I'm not E. coli, I'm really staph. They did the wrong one. So what that means is you want to work slowly, and you really want to look at what the tube labels are before you set anything up. Okay? Now, we're going to be doing this lab in pairs, so we're going to be working together. So, for example, here's my MSA. This is mannitol salt auger. And what you're going to do is you're going to draw a line down the center. Okay, so you're going to take your plate, and you're going to draw a line down the center. And then your lab partner is going to take that same plate. So we should have an MSA for partner 1 and an MSA for partner 2. Okay, I'll just put P1 and P2. Okay, and what you're going to do then is you are going to decide which organisms are yours to set up, and your partner is going to decide which organisms are theirs to set up. So with that in mind, for example, I have E. coli and I have Proteus. So I might decide I will do E. coli and Proteus on all the media. My lab partner will do the 2 staffs on all their media. So you're going to divvy, okay? Now, if I have, for example, Proteus, I'm going to sterilize my loop. I'm going to take my loop and gently scrape some Proteus. Do I need a whole lot? No, because this is dry crow's. Couple of things to keep in mind. Remember, this is my slant here. When I go in with my loop, I am not digging. I'm not being rough. I'm very gently scraping upwards along the surface, and I will pick up a little bit of paste. And as soon as I see that paste, I'm done. Remember, I don't need a whole lot. It's kind of like when we did our slides. So what I'll do then is once I've picked up that bacteria, I'll come over here and I'm going to do what's called a fishtail streak. So I'm going to take my loop and I'm going to streak down on the plate. That's my first streak. And then I just take the loop and go back to the top. I'm not re-inoculating. I have enough bacteria, so I scrape down. And then with the same loop, I just come here and I zigzag. So it's called fishtail. All right? And you can spread it out a little so that you get a little bit bigger of a swipe. But the one thing you want to be careful of, you do not want to go to the very edge. And the reason for that is there's always a little moisture around the edge of the plate. If I get in that moisture, it's going to swing around the entire plate, and then it might possibly contaminate the surface next time and make it harder to tell my results. The other thing is I want to be careful to not cross over into the other section, because then I'm going to be affecting the neighboring bacteria. All right? So when you're labeling these, remember you can write directly on your plate. You'll do a line. Let's say you put Proteus, so you put Proteus here, you put E. coli here. Remember, so if I'm labeling Proteus here, when I flip them over, Proteus is now on this side, not on this side. So you want to make sure you're putting the right organism in the right place. Everybody with me on that? So all of us are going to be setting up one mannitol salt auger with 2 organisms. You and your lab partner set with 2. One nutrient auger with those same 2 organisms. One McConkey auger. And then one EMB, all right, with those same 2 organisms. So whichever ones you pick, those are yours for the day. All right, everybody got an idea of what we're doing? Couple of hints I will give you. Be sure to flame sterilize longer than normal between your different organisms, because you will get carryover. If you're getting paste on your loop, you got to get rid of all of that bacteria before you pick up your next sample. The other thing, I've had students try to do this in the past. They go, oh, it's the same bug. So I'll just get a big loop and I'm going to go here and here and here and do them all at once. Do not do that. There are different ingredients in each of these and you will get carryover. So let's say that bacteria is inhibited on this one and I just streaked it. I'll take that inhibition to the next plate and you won't grow when you should. So you'll have to flame sterilize between the different media. All right? Everybody got the game plan? Okay, so let's go ahead and get started then. Get your game plan started. Figure out which organisms you're going to set up. If you need media, let me know. You can have it at your desk, but if you don't, we're on
Oh, you won't go anything. I would have heard.
Yeah. You're right.